Assalamu alaikum wa rahmatullahi wa barakatuh. Hello, grade 9 advanced biology students. Welcome to your term 3 revision session. Hope you are all doing well and feeling motivated as we approach the final exams. Today's session is designed to help you recall key concepts, clear any doubts, and boost your confidence. Make sure you have your notebooks ready and let's review smartly, not just hard. Let's begin our journey through the amazing world of biology, one concept at a time. Myself, Miss Abeda Peerzade from Madhab Girls School Cycle 3, Fujairah. So as you can see, the EOT, that is end of term, or you call it as Heikel. So here you will have 25 multiple choice question. Each question will have 4 marks. The time has the same 2 hours. It is a paper-based exam. 100 marks questions, 100 marks for the exam. And again, I would like to mention that it's all 25 questions are multiple choice questions. Students sometimes ask whether we will have any writing question. So you don't have anything writing, you have multiple choice questions. So let us begin with the first learning objective for today's session. Question number 1 is from page number 207. Explain the major events that occur after fertilization. Okay, that means we assume that the fertilization has already taken place. So you must know first what is a fertilization means. Okay, fertilization is the union of the egg and sperm. And when this union happens, a diploid cell, that is zygote, is being formed. All right. Now, the fertilized egg, I'm not just saying egg, I'm saying fertilized egg is called as zygote and moves through the oviduct with the help of cilia and smooth muscle contraction. This is how the development of the embryo begins after the fertilization. So there is a step-by-step developmental stages. First step. About 30 hours after the fertilization, the first zygote divides for the first time. By the third day, remember the sequence, 30 hours, then third day, the embryo becomes a morula. Now you should know this. What is morula? Very important. Okay. This can be asked in exam. Morula is a solid ball of cells. How many cells? 16 cells. A solid ball of cells is called as morula, which is formed by third day, okay, and enters the uterus. On the 5th day, the morula changes into blastocyst. Now you should know what is a blastocyst, okay? Blastocyst, which is a hollow ball of cells. Blastocyst is a hollow ball of cells. Now, this blastocyst attaches to the wall of uterus, which is called as implantation or uterine wall, or it is also called as endometrium, around the 6th day and is fully implanted by day 10. So blastocyst looks like this. This is called as inner cell mass of blastocyst. If this inner cell mass divides into 2, then there is a possibility of 2 identical twin formation. All right, so let us see the possible questions on this topic. When does the first mitotic division of the zygote occur after fertilization? So first mitotic division, the zygote divides by mitosis, that starts around 30 hours. When does the blastocyst attach to the endometrium? Around 6th day. What is the name of a solid ball of 16 cells? that is morula. What part of blastocyst will eventually become an embryo? That is the inner cell mass. Now, when you solve any question, you need to read the question very well. You need to focus on the keywords to find the exact answer. So this question had been asked in previous exam. How many chromosomes are there in the not fertilized human egg? Don't just focus on the egg. Okay, read the question completely, that it says a not fertilized human egg. Yani fertilization has not happened. Egg is egg with 23 chromosomes. So that's why the correct answer is 23. If they say fertilized egg, then you can say 46. But they say not fertilized human egg. Please read the question well. Otherwise, if you will focus on fertilized and egg, you may end up selecting 46, but don't do that. Please read it well, completely, calmly. Not fertilized human egg, that is 23 chromosomes. Which of the following describes the proper sequence of development? That is, first zygote is formed, it gets converted to morula and then to blastocyst. This is the correct order. In which stage the embryo implanted in the endometrium of uterus? Implantation. Blastocyst implants itself into the endometrium. When the embryo is about to leave oviduct and enter the uterus is called as blastocyst. Now, question number 2, explain how the process of meiosis ensures the reduction of the chromosome number. Straightforward it is. How does meiosis help in reducing the chromosome number? Meiosis occurs in the reproductive structure of organism that reproduces sexually, forming haploid gametes. This is the importance of the meiosis. It's a type of a cell division in which the main focus is, or the main purpose is to form the gametes, male gametes or the female gametes. Now, it reduces the chromosome number by half. This is very important. It reduces the chromosome number by half through the separation of the homologous chromosome. How the chromosomes number is being reduced to half? By separation of the homologous chromosome. A cell with 2n chromosomes will have gametes with n chromosomes after meiosis. Yani the diploid gets converted to n, that is haploid. The sexual life cycle in animals involves meiosis. Okay, meiosis is a type of a cell division which happens for the sexual reproduction, which produces the gametes. Gametes, what are gametes? Sex cells. For example, in the male, we have sperm, and in the female, egg. Those are called as gametes. When gametes combine in fertilization, the number of chromosomes is restored. When does restoring happens? After fertilization. Okay. The chromosome number is retained to 46 only when the fertilization happens. Okay. Otherwise, in meiosis, the chromosome number reduced from 46 to 23. It is the fertilization process which restores the number. Meiosis is the process in which chromosome number reduced to 23. Let us see the questions based on this learning objective. What happens to the number of chromosomes during meiosis? It reduces to half. Meiosis reduces to half. Remember. Which process produces gametes? That is meiosis. So when you will go through various kinds of questions, your concept of meiosis will become very strong. Which process ensures to ensure to restore the chromosome number in animals for the next generation? Now, what is being asked You have been asked a process. A process in which the chromosome number is being restored. That is fertilization. Which event during meiosis reduces the chromosome number by half? What is that event when the chromosome number gets half? That is the separation of homologous chromosomes. Okay, repeat it twice, thrice. Separation of homologous chromosomes creates the haploid. Which process is shown by letter A? Now, this is on page number 236, figure number 15. Wherever they have mentioned figure, there are possibility that the figure may come in exam. So, which process is shown by the letter A? So, here is a letter A. What do you see before letter A? There is a sperm and egg. Okay. So, if they unite, what would happen next is a fertilization. Now, coming to the next question, which one can be denoted by 2n set of chromosome? Now, 2n itself means diploid. All right. So that is zygote. Male gamete and the female gamete is only denoted by single letter N. Okay. So that is zygote. Question number 3 on page number 201. Explain how hormones regulate the male reproductive system. It's talking about male reproductive system to include testosterone, GnRH, FSH, and LH. All right. So let us understand the male hormones. Testosterone is produced in the testes. It's the male hormone and is essential for sperm production. See the connection? Testosterone is produced in testes and it helps to produce the sperm. It also causes male secondary sexual characteristics like facial and chest hair, broader shoulders, more muscles, and deeper voice. We are talking about the testosterone hormone, where it is produced and what does it cause. The hypothalamus releases GnRH. Remember hypothalamus with GnRH. Hypothalamus releases GnRH, which is called as gonadotropin releasing hormone. In short, we call it as GnRH. It is released by hypothalamus. Look at here. This is a hypothalamus which releases a hormone called as GnRH. Now, what does it do? GnRH acts on the anterior pituitary gland. So we have a pituitary gland over here divided into two lobes, anterior and posterior. Okay, so we are focusing on the anterior pituitary gland. Now, what does GnRH do? GnRH, which is released from the hypothalamus, acts on the anterior pituitary gland to produce FSH, that is follicle stimulating hormone, and LH, that is luteinizing hormone. So what GnRH does? It acts on the anterior pituitary gland to release FSH and LH. Now, how this FSH and LH influence, we will see now. FSH and LH travel into the blood to the testes. This is a target organ, target gonads. Testes are the male reproductive organs. So this FSH and LH are going to act on the testes. In the testes, FSH helps making sperm. It helps for sperm production, and LH triggers the testosterone production. You need to understand how FSH and LH work in male. FSH helps to make the sperms. LH helps to trigger or regulate the testosterone production. Understand? LH will trigger the testosterone. FSH will help to make the sperm. Clear? We will see the example questions. Which hormone is also called as a male hormone? That is testosterone. Male hormone, testosterone. Which hormone stimulate testes? Okay. Which hormone stimulate the testes to secrete the testosterone? That is LH. So LH will stimulate the testes to make testosterone. Okay, so there is a connection between LH and testosterone. Keep it in the mind. Which of the following is an effect of FSH? Now we already know that FSH helps the testes to make the sperm, right? So FSH and sperm production. Remember this. FSH with the sperm production, LH with testosterone production. Now, which of the following hormone influence the production of testosterone hormone? That is GnRH. So GnRH influences the production of testosterone. Moving to the next learning objective. This is from page number 201. Okay, we are in the same question, but we will see a little more example. Which of the following sentence is not correct for testosterone? Okay, now this is very important for you all to read the question wherever not is mentioned. This is very important. Otherwise, you will just see the testosterone and you will select one of these. Always remember when you answer the question, each question has 4 marks. One wrong answer can lead to -4 straight away. So read the question well. What is not correct about testosterone here? So it causes ovulation. Ovulation is seen in females and not in the males, right? So testosterone doesn't cause ovulation. FSH and LH hormones are released from anterior pituitary gland. Here, this is the anterior pituitary gland. From here, FSH and LH are released. Which hormone is secreted from hypothalamus? Hypothalamus releases GnRH. Which is the following is not correct? Please circle your keyword, highlight your keywords. Okay, something which is wrong over here. The level of LH and FSH is regulated by a positive feedback mechanism. No, that's a negative feedback mechanism. Okay. So what you have to do, you have to go through all the options to come to the conclusion for your correct answer. For example, GnRH activate pituitary to release LH and FSH. Yes, that's correct. FSH stimulate sperm production. Yes, it is correct. GnRH release from hypothalamus to activate the anterior pituitary. That is also correct. So what is wrong about FSH and LH? That is regulated by positive. So that's wrong thing. Moving to the next questions. Few more example questions. Now, which of the following is an effect of FSH hormone in testes? Now you have to read it completely. Don't just read FSH and try to find the answer from this. Read, is it testes or ovaries? Now they mention testes, so you're thinking about male hormone. Or how does a FSH hormone affect the testes? So they help in sperm production. So the answer is the production of sperm. All right, moving to the next question. In the picture below, which of the following hormones stimulate the production of FSH and LH? That is GnRH hormone. You need to read like what is being asked. Are they asking you the gland or hormones or what? So they asked about hormones. Which hormones stimulate the production of FSH and LH? That is GnRH hormone. Which of the following hormones stimulates the gland? Look at this. Which of the following hormone stimulate the gland shown in the image? This is the image. Below that, regulate the testosterone, that is GnRH hormone. Moving to the next learning objective from page number 204, table 1. So there is a possibility of this table diagram to come in the exam. Explain how hormones regulate the female reproductive system to include estrogen, progesterone, LH, and FSH. Okay. So what you should know from this table, that this is the table which displays the menstrual cycle process. So menstrual cycle is divided into 3 phases: flow phase, follicular phase, and luteal phase. Okay, this is the order. You should know the days of each phase, duration of days of duration of each phase. Then you should know the hormone levels. Look at this order: FSH first, second LH, third estrogen, fourth progesterone. Remember, now in exam, you may not have the same color which is there in your textbooks. Please keep in mind. Otherwise, you will think that in our textbook, LH was shown in yellow color. So you are expecting the yellow color line must be for LH in exam. No. Look at the order: FSH, LH, estrogen, progesterone. Let them give any color to them. All right. And you already know what is the level of hormone in which phase. Okay. Flow phase, all the hormones are down. Follicular phase, you can see FSH is increasing gradually, then estrogen, and at the end of follicular phase, that is LH, which is at surge or increase, causing the ovulation. In the luteal phase, that is the last phase where the progesterone is very high, only in the luteal phase, right? You should know that this is a follicle. So follicle maturing happen in the follicular phase. Then at the end of follicular phase, the rupture happens, that is called as ovulation, where the egg is released, follicle is ruptured, follicle is broken down to release the egg. What happens to this broken follicle? It gets converted to corpus luteum, which release the progesterone. That's why the level of progesterone in the luteal phase is very high. Let us understand. So as I said, in the first phase, look at the word flow. We will highlight this word flow. Flow yani flowing. So in the menstrual cycle, the first phase is called as a flow phase. How do we describe the flow phase? It is the shedding of blood, okay, mucus and epithelial cell from endometrium. That's the wall of the uterus. The length of the menstrual cycle can vary or differ, but it typically lasts about 28 days. The cycle can be divided into 3 phases: flow phase, follicular phase, and luteal phase. All right. So flow phase lasts for 1 to 5 days. At the beginning, when estrogen level is low, the anterior pituitary increases the secretion of LH and FSH. LH and FSH stimulate the follicles in ovary to begin maturing. The size of the follicle increases. Maturing follicle or growing follicle releases or secrete the estrogen and small amount of progesterone. The secretion of the estrogen and progesterone inhibits means prevents the pituitary for secretion of LH and FSH. By negative feedback mechanism, okay, about day 12, high amounts of estrogen causes the pituitary gland to release a surge of LH. LH increases. Surge yani increase. Surge means increase. Surge of LH. Rapid release of LH causes the follicle to rupture. Rupture means to break. And ovulation occurs. We will see some example questions. Which of the following number represents the estrogen hormone? Now let me make it bigger. Estrogen. So I told you to remember the order of the hormones. So first is FSH, second LH, third estrogen, and fourth one is progesterone. So they asked you about the estrogen. That's number 3. This is estrogen. Okay, which is colored in yellow. So don't think about the colors. Colors can be any colors. They are mentioned, may not be the same color as mentioned in your textbook. I'm repeating it again. Just remember the order of the hormone. First FSH, second LH, third estrogen, and fourth one progesterone. So number 3 is your estrogen. Which of the following numbers represent LH hormone? LH is number 2. Which of the following is not a menstrual cycle phase? We know that there are only 3 phases. So gametogenesis is not the phase. In which phase of the menstrual cycle the progesterone level is very high? That's the last phase, luteal phase, where the progesterone is very high. So you can see progesterone is denoted over here by number 4, where it's very high in luteal phase. All right. So you can see page number 204, table number 1. A few more questions on the same learning objective. How many days? Stage 1 is 1 to 5. Follicular phase of menstrual cycle is represented by letter B, follicular phase. So you can expect, if the diagram is being asked, it can be about the hormones, hormonal level which are represented here in the graph, or they may ask you about the names of the phases. So B is follicular phase. What causes ovulation? That is LH. Remember, ovulation with LH. Surge of LH, increase in the hormone LH, causes the rupture of the follicle and the egg is being released. This process is called as ovulation. Which of the following sentence is not correct? Okay. Now, for that, you need to read all the options. A rapid decrease in the LH causes ovulation? No. The surge of LH or increase in the amount of LH causes ovulation. This question was asked in previous exams. Which of the following number represents the estrogen hormone? See, they had asked which is the estrogen hormone. Here the names are not mentioned, but you know that first is FSH, second LH, third estrogen, and fourth one progesterone. So look at the color of 3. Okay, this is your number 3 is your estrogen. Learning objective number 5, define the terms gametes and gonads. Again, meiosis produces 4 sex cells which are called as gametes from one parent cell in testes in males and ovaries in females. In males, sperms are produced daily from primary spermatocytes starting at puberty and continuing throughout the life. In females, all eggs begin development even before the birds. Primary oocytes are formed with replicated genetic material. Meiosis in female stops or pauses before the first division is completed and resumes 1 egg per menstrual cycle. The first meiotic division in female results in a large oocyte and a smaller polar body which breaks down or which disintegrates. It is very important. There are 2 meiotic divisions, right? Meiosis 1 and meiosis 2. At the end of first meiotic division, polar body is formed and large oocyte is formed, okay, which is called as a secondary oocyte. At the end of second meiotic division, what happens? It occurs only if the fertilization happens. If the sperm unites with the egg, what happens? The zygote is formed. And here the second polar body is formed. and the zygote is formed. Second polar body, which is smaller comparatively to zygote, which disintegrates. Okay. So first meiotic division, first polar body. Second meiotic division, second polar body and zygote. Remember this. Okay. So we are talking about the gametes and gonads. We just have two gametes. Male gametes are sperm and female gametes are eggs, where the egg cells are produced in the ovary. That's the female gonad. Which of the following process give rise to the formation of sex cell? Sex cell production takes place because of the process meiosis, a type of a cell division which reduces the chromosome to half. Now, haploid is sperm and egg cells are haploid. They have 23 chromosomes, right? Male gonad testes produce. So male gonad is testes and female gonad is ovary. Remember. So male testes produce sperms. These questions were asked in the previous exams. Which in the following mentioned to meiosis occurs only in the reproductive cells? Meiosis happens only for the production of sex cells, that is the sperm and the egg. What results from the first meiotic division? Polar body. Now here they haven't mentioned first polar body, second polar body. No way, no problem. Development of the polar body is a correct answer. Rest you can see sperm formation, ovulation, development of a zygote. That's wrong. Coming to the next learning objective, identify the structures and their functions of the female reproductive system to include ovaries, uterus, vagina, and oviduct. Okay. This is about the female reproductive system. These are the two diagrams mentioned, figure 4 on page number 202. So we are expecting a diagram to be asked in exam for 4 marks, right? So you should be knowing the labels correctly. Now, you see this is called as a tube-like structure, okay, which carries the zygote. Then fertilization also takes place over here. Okay, it gets converted to morula and blastocyst. All this development happen in the oviduct. Now, if you see this blue round structure, that is the egg which is released from the ovary. Look at this. This is a broken follicle. Rupture any broken. Follicle ruptured and the egg has been released. Now, what are these? These are all the follicles. Okay. And inside the follicle, there is a egg. Now, this light yellow colored egg-shaped structure, oval-shaped structure, this is ovary. And this is the broken follicle, which is over here, which is called as a corpus luteum formation. These are all corpus luteum. Okay. Now, in the exam, if this diagram comes, you have to be very careful where these arrows are. It's very close. So you should know the position of the ovary. Same diagram will come in exam. And which one is corpus luteum. In this diagram, you should know this portion is vagina. At the beginning or the entrance of the uterus is the cervix. Then this is uterus where the embryo develops or the baby develops. This is ovary and the tube-like structure is oviduct. Now let us understand some diagram labeling questions. What does C denote? C is a oviduct. Here D, now look at this, D is just the egg. Okay, this is a released egg. Now, follicle, growing follicle. So letter C is a growing follicle. You can see this is a fully matured follicle, which is not correct. So estrogen and progesterone are female hormones. Baby develops in uterus. Oocytes are immature egg cells, but egg cells are produced in oviduct. This is wrong information. Egg cells are produced in ovary. Now, which number in the picture represents the ovary? Again, I said, look at this. Number 4 and number 3 are very close. You have to be very much sure about what you are answering. Number 4 is ovary and number 3 is corpus luteum. Not mentioned, not a main structure of a female. So first 3 are female reproductive structures. Epididymis is present in the male reproductive structure. They asked about the ovary. Here, number 5 is the ovary. A represents cervix. Some students, they make a mistake in identifying A as uterus. Letter B is uterus and letter A is the cervix. Keep in mind. Now, learning objective number 7, identify the structures and their functions of the male reproductive system to include testes, epididymis, scrotum, seminal vesicle, vas deferens, and prostate gland. All right. So here also you should be able to identify different labels in male reproductive system. Male reproductive glands are called as testes. You can see there are 2 testes. This is a testis and this is testis, denoted by letter F. They are located outside the body cavity in a pouch called as a scrotum. The temperature lower than 37 is required for the development of the sperm. That's why they are outside the body. Our human body temperature is 37. So for the sperm production, lower temperatures are more suitable. This is the reason why testes are outside the bodies. Now you can see this is testis. On the top of the testes, there is a coiled tube called as epididymis, where the sperms are stored and matured. In the testes, sperms are formed, but they are stored and developed matured in the epididymis. Epididymis continues into a long, thin tube on either sides, which is called as vas deferens. Now, vas deferens continues here into a seminal vesicle. And then you have this prostate gland. Below that, these are tiny bulbourethral gland. Then this continues into a common tube, okay, which carries the sperm and the urine both, and they are excreted of the organ which is called as a penis. All right. Let's see the example questions. The tube that carries both semen and urine outside the body through the penis is called as urethra. Now, epididymis, which number represents? Look at this again. Number 2 and number 3 are very close. So here you need to observe the diagram very well. Number 2 here is testes. Number 1 is a penis. Number 2 is testis. Number 3 is epididymis. So you have to be careful where the labels are very near to each other. Vas deferens is D. You can see this is testes, epididymis, and this is a vas deferens. C, what is a C? C is, what does C labeled as? Testes. So inside the testes, what happens? Sperm cell production happens. Next question is differentiate between the terms zygote, embryo, and fetus. Okay. So here we can learn about extra-embryonic membranes, okay, and their functions. So there are 4 extra-embryonic membranes: amnion, chorion, allantois, and yolk sac. And also we will talk about the placenta. So here we are differentiating zygote, embryo, and fetus. Okay, we are talking about question number 8 and 13 together. So let us first see what is amnion. Amnion over here, you can see this is a thin membrane around the embryo. It is a thin layer and form a sac around the embryo. Look at this word sac. Sac around the embryo. This sac holds a fluid called as amniotic fluid. What is the function of this fluid? It protects, cushions, and insulate, helps to maintain the temperature or regulate the temperature of developing embryo. Chorion and allantois together. Now, chorion is the outer thick layer. And allantois, you can see this green structure, which is attached to the embryo. Okay. So this chorion and allantois together. So you can see this forms the tube, which is called as umbilical cord. And also it extends into placenta. So chorion and allantois together contributes to the formation of placenta. Yolk sac, you can see yolk sac like a egg yolk, which is yellow in color. You can see this is a yolk sac. Very distinct function it has. In humans, the yolk sac serves as the first site for the red blood cell formation. Now, a little more about placenta. As an embryo develops, the chorionic villi, this is a chorionic villi. As an embryo develops, the chorionic villi begin to grow into the uterine wall. Nutrients, oxygen, and waste diffuses across the maternal and fetal blood vessels and carried to and from the fetus through the umbilical cord. The placenta provides food and oxygen. Please remember this, the function of placenta, and removes the waste from the fetus. The placenta contains tissue from both mother and fetus. Now, what are the 4 extra embryonic membranes? So you should select the correct answer: amnion, chorion, yolk sac, allantois. Not the extra embryonic membrane, umbilical cord. What is the sac around the embryo? Amnion. Immediately around the embryo is amnion. Now, which structure connects to the fetus and provide the food and oxygen? That is umbilical cord. Okay. Now, you may say it is placenta, but read the question well. What is the name of a structure that connects to the fetus? Connected to the fetus, which supply this, so that is umbilical cord. What is the function of a yolk sac? To produce the red blood cell for the fetus. Some more questions. Now, amnion. Amnion is number 2. Allantois, it's a green smaller structure. You will have the colored pictures in the exam as well. You will see the same extra embryonic membrane diagram which is there in your textbook. So allantois is denoted by number 5. Yolk sac, you can see these all structures are very closely. Again, I am asking you to be careful when you answer the question. This is number 6 is for yolk sac. Chorion, very easy to identify, number 1. Question number 9. Sketch the specific events that occur within each stage of mitosis and cytokinesis. Page number 228, figure number 6. We are expecting this figure or individual phases would be asked in question. So we know that mitosis have 4 phases. Okay, we denote it with the code PMAT, or we call it as PMAT. All right. Now, so this is a prophase, metaphase, anaphase, and telophase. What about this? This is the interphase. So Mitosis is a type of a cell division, okay, which helps in the growth and repair, which makes the cell to restore its chromosome number, that is 46. Prophase. Chromatin condenses to become chromosomes. So you can see here, you cannot see the chromosome condensed. Okay, it's a relaxed form of chromosome in the interphase. But moment prophase starts, you can see this nuclear material or the genetic material get condensed and appear as X-shaped structure, that is chromosome. So for the first time, you see the chromosome in the prophase. Then the metaphase, meta yani middle or central line or chromosome align in the center, or you can say they are lie in the equator. Chromosomes align on equator. So that's a metaphase. So sister chromatids are pulled by motor protein along the spindle apparatus towards the center of the cell and line up in the middle or equator of the cell. Now, not necessary, I have repeated this again and again, but if you don't find the cell in this position or you find this line as horizontal, that's okay, absolutely okay. That is also a metaphase. The cell can be represented in a vertical or it can be a horizontal line. But if all the chromosomes you see aligned on equator or in the middle line, that is metaphase. Anaphase, ana, remember A for ana, A for away. What exactly happens over here? The chromatids are pulled apart. You can see these are the chromatids. They are moving away from each other. Sister chromatids are separated into two identical chromosomes. So you can see this is the anaphase. And finally, the telophase is very easy to identify, where the genetic material of the nucleuses are separate. This is the last phase. Telo means end. Okay. Last phase in mitosis. Chromosome decondenses. Two new nuclear membranes begin to form and the nucleoli reappear. Now, next questions. Okay, it is the same learning objective, but few more questions we will see. In what stage the cell cycle does the cell's replicated genetic material separated? So, how to choose the answer? Read the question well. They are asking the stage. Okay. Of what? Of cell cycle. You know that there are three stages of a cell cycle: interphase, mitosis, and cytokinesis. So, when does the genetic material is separated? Genetic material is separated in the mitosis. What exactly happens in mitosis? The nucleus gets separated. At the end, in telophase, two nucleus are formed like this. Stage we want. Stage of what? Mitosis, where chromatids are lined up at the equator of the cell. Whether the diagram is given or not given, if you find the keyword, well, you can select the answer confidently. See, these are the keyword for metaphase: equator, lined up in the center, lined up in the middle. Stage of mitosis we want, and the picture is given. So this is the first phase of mitosis, where you can see the condensed chromosome. They are not lined up in the straight line in the middle, or they are not, nucleus isn't separated, all right? Or the chromatids are not pulled away. This is the prophase. Stage of mitosis. This is the last phase in the mitosis, that is a telophase, where the nuclear material is divided. Few more questions. Anaphase we need to find from this. This is anaphase A, telophase, prophase, metaphase. Now find out the metaphase. Very easy by now. You should be expert to find out which one is metaphase. This is what I was saying. Not necessary the cell would be shown vertically, it may be horizontal also. So what is A in the mitosis? A is interphase. So prophase, metaphase, anaphase, telophase, and this is interphase. When you can see the chromosome for the first time, so here you can see the nucleus where the chromosome is relaxed. Chromosome are seen in a condensed form in prophase. That is denoted by C. Learning objective number 10, compare and contrast mitosis and meiosis. Okay, including their stages, outcome, contribution to genetic variation. It's on page number 240. So the basic differences between the mitosis and meiosis. We all know that both are type of cell division. Mitosis divide only one time. Meiosis has two divisions, meiosis 1 and meiosis 2. Then DNA replication occurs during S phase of interphase in mitosis. But DNA replication occurs once before meiosis 1. Crossing over or synapses. Look at this. This is another word for crossing over, synapses. So you can be asked only about synapses. So don't think that you haven't heard this word. Synapses is the other word for crossing. Of homologous chromosome does not occur in mitosis. It's not needed. But in meiosis, crossing over or synapses of homologous chromosome occurs during prophase 1. Remember this. Crossing over, prophase 1. Crossing, prophase 1. Two identical cells are formed and four haploid cells formed which are genetically different. And these two diploid cells are genetically identical. Mitosis occurs only in the body cells, whereas meiosis only occurs in the reproductive cells to produce gametes. Those are sperms and eggs. Now, let us see the example question. Which of the following is true about meiosis? Occurs only in the reproductive cell. Not correct about meiosis. It doesn't double the number of chromosomes. Rather, it reduces to half. Correct regarding mitosis. See, there are some students. How do they do the mistake? I will tell you. They do not read it well. In a hurry or in a stress of exam, sometimes students read meiosis as mitosis and mitosis as meiosis. That is why you need to write these words when you are studying with the correct spellings, and you need to pronounce it very clearly. Mitosis, and this is meiosis. All right. Mitosis, only one division take place. And you should also see what is being asked. That's what's correct. What's true in meiosis? True, and what is correct about regard to meiosis? Two sets of division take place. That is meiosis 1 and meiosis 2. What is formed in meiosis? Four haploid cells are formed. What is not the importance of meiosis? Growth and repair. Because in meiosis, gametes are formed. Growth and repair is the importance of mitosis. Which of the sentence is correct for mitosis? That it divides only once. Which of the following is the importance of mitosis? That is the function, growth, and repair. If you will go through all this material, you will be really, I'm saying, you will be expert in understanding the concept. Question 11, identify the similarities and differences within homologous chromosome. So, To answer the questions on this learning objective, you should first know what is homologous chromosome means. Homo yani same. Okay, homo means same. Chromosomes that make up a pair, so you can see there is a pair, one from each parent, are called as homologous chromosome. What is more specific about homologous chromosome here, even if you will see the diagram, you can make out that they have same length. Look at the length is same. They have the same centromere position. Look at this centromere at the same level. They carry the genes that control the same traits. So these lines are for the genes which control the same traits. Homologous chromosomes are physically bound together during the synapses in prophase 1. Synapses, again, I would say it's a crossing over. Which structure holds the sister chromatids together? That is centromere. I will tell you what the students make mistake over here. You can see here it is centromere and next option is centriole. And now in exam, the student get confused. What is the correct answer? Is it centriole or is it centromere? Now there is another word called as a telomere, which is also associated with the chromosome. The ends of the chromosome is called as the telomere. The center, at the center where the two sister chromatids are attached, is called as centromere. Remember centromere, okay, which holds the sister chromatid together. Which phase of the meiosis? When the synapses occur between the homologous chromosomes, that is prophase 1. What is B here? B is a sister chromatid. B is a sister chromatid. From the same chromosome, two sister chromatids alike. Now, same length and centromere position, they are homologous chromosome. Now, one thing you should know that in this diagram, B is sister chromatids because they are alike. What about C? C is called as non-sister chromatids. They are from two different chromosomes, right? So they are non-sister chromatids. Now, question number 2. Define fertilization as the process that restores the chromosome number by combining the egg and sperm to form a new individual. This is on page number 206, figure number 7. So the possibility of the diagram coming in exam. To answer the question on this learning objective, first you should have the concept clear for fertilization. What is fertilization? This is a process of sperm uniting or joining or fusing with an egg, which means the sperm nucleus fuses with the egg nucleus and makes a zygote. Fertilization usually occurs in oviduct, which is also called as fallopian tube. So where does fertilization take place? In oviduct. Sperm and egg are both gametes. They are haploid, denoted by single letter N. And each normally have 23 chromosomes. Fertilization is a process which restores the diploid number, 46 a chromosome. Single sperm cannot penetrate the plasma membrane of egg. Single sperm, you can see here, there are many sperms trying to penetrate the egg plasma membrane, but only one gets success and enters inside. So you can see the head of the sperm has entered inside the egg. So now if the single sperm cannot penetrate the plasma membrane, then what exactly is needed? is written over here. The acrosome, it is the tip. You can see the acrosome over here. Acrosome is the tip of the head. All right. Acrosome is the tip of the each sperm cell contains the enzyme lysosome. At the tip of the head of the sperm, there are enzymes which are called as lysosome. That lysosome weakens the plasma membrane surrounding the egg. So this is the cumulative effort of all the sperms surrounded on the plasma membrane, which release a lysosome to weaken the plasma membrane. Lysosome from hundreds of sperm make the plasma membrane enough weak that one sperm can penetrate the egg. What helps the sperm to penetrate the plasma membrane? That is the enzyme called as lysosome of the part which is called as acrosome. What is this process? Moment you see this picture, you should be confident enough to say this is the diagram of fertilization. Human female egg have 23 chromosomes and human male sperm has also 23 chromosomes. After fertilization, look at this word, after fertilization, the zygote have, now you may say, where is the number 46 here? So I am confused to answer this question because zygote, I know that it has 46 chromosomes. But 46 is not mentioned over here. Please go through all the options well. 23 chromosome, wrong. 30 chromosome, wrong. 44 chromosome, wrong. Come to this option, 23 pair of chromosome. 23 pair means total 46 chromosome. Now you can expect such indirect questions as well, though it looks very easy, but it can confuse the students. All right. So this question number 14. Explain how hormone regulate the male reproductive system to include the testosterone, GnRH, FSH, and LH. All right, here this is a question number 12. Question number 13 is coinciding with question number 8. So we have already discussed about it. So the production of FSH and LH hormone is stimulated by GnRH hormone. Hypothalamus, this is hypothalamus, release GnRH. LH, which is also called as luteinizing hormone, LH. What does it do? LH, I told you to remember LH with the testosterone. It promotes the testosterone production. What is the function of follicle stimulating hormone in males? They didn't say females. Please read it well. FSH in male. So males have testes and FSH helps to produce sperm. So it promotes the sperm production. Summarize the development that occurs during each trimester. Page number 210, figure number 11. Now, please do not lose your marks in this question. Definitely, you will be asked a diagram to identify the weeks of fetus. Or they may give you the weeks and they may ask you to identify the diagram from these 4 pictures, any 4 of these. Again, I am reminding you that these pictures, as they are shown in the textbook, in exam, you might not get them all in order. They may be rearranged. For example, the 12-week, maybe the first, second is 5 to 6, third one is 9 weeks, and then 7 to 8, like that. Okay, it's very easy to differentiate between them. Now, if you will look at this first picture, 4 weeks, you can see and identify it very easily. So the organs You cannot differentiate them, the face and hands and legs, you can't differentiate them. So 4 weeks baby. So look at the progression after 4, 5 to 6. The second diagram or the second figure, look at this, it's like a shrimp, right? A tail-like structure it has. It's 5 to 6. Remember, first one is deformed or it is not formed well. Second one, like a tail it has, 5 to 6 weeks. Third one, you can see, you can spot this eye in 7 to 8 weeks. Why I am saying you this? Because it would be easy for you to identify. It will easy for you to score the marks. 7 to 8 weeks, you can spot the eye. 9 to 10 weeks, you can see the bump on the head. Okay, now these two pictures look very similar. But what's the difference here? 9 to 10 weeks, baby, the look at this head is bumpy or like a swollen. 12 weeks, baby, the picture is very clear. The head is straight. All right, well developed fetus. All right, coming to the some example questions. Which number represents 7 to 8 weeks baby? So we have, look at this. They are not in, here you can see they are in order, but in exam you may not find them in all order. Now we have 4 weeks, 5 to 6, 7 to 8, 9 to 10, and 12 weeks baby. So what do you see in the 7 to 8 week baby is the eye. You can see, you can focus on the eye. That's number one. 9 to 10 weeks baby. This is 9 to 10 weeks baby. Number 3 with the bumpy head. 4 weeks is very easy. That's 2. And 12 weeks is a well-formed baby. That is number 5. Now, talking about the trimester-wise development, which trimester where the human embryo is developed is called as fetus. Fetus is the embryo which develops or which completes 2 months of its age. So that comes in the first trimester. And then which trimester? You can see the fingerprints developed. That is first trimester again. On page number 210, this talks about the first trimester mostly. Brain development happens in the third trimester, which is on the next page, actually, 211. Which hormone is released during the first week of embryo development? Very important question. This is a hormone, human chorionic gonadotropin, which is released, okay, during the first week of embryo development. With this hormone, even the pregnancy has been confirmed. Learning objective number 16, page number 224, figure number 1. Now, in this learning objective, you will be given a problem-based question where you need to find out the surface area to volume ratio. To answer this question, what you should know, you should be able to calculate and determine the surface area. So we need the formula for the surface area and volume. And then finally, we will calculate the ratio. You will be given any of this cube, okay, either 1 or 2 or 3 or 4. Any of the cube would be given. Now for the cube 1, the ratio is 6:1. For cube 2, it is 3:1. Cube 4, yani the cube which has the side 4 micrometer or 4 centimeter or millimeter, whatever. So it is 2:1. Now how to calculate? See, the surface area formula is length into breadth into number of surfaces, okay, for the cube. Now, say for example, this cube has got a side 1 micrometer. Because it's a cube, length and breadth and height would be all same. So length 1 micrometer, breadth 1. This number is constant for the cube. 6 sides are there for the cube. So 1 into 1 into 6, that gives you 6. Okay, 6 centimeters square, that is the surface area. And the formula for the volume is length into breadth into height. All same, so it is 1 centimeter cube. 1 times 1 times 1 is 1 only. While the largest cube has the ratio, if we talk about this large cube, it has got a side 4. So substitute this value 4 into the formula. Length into breadth into number of sides. 4 times 4 times 6 is 96. And 4 times 4 times 4, that is volume, is 64. Now, in the options, you may say the answer is not there because you got the answer as 96 is to 64. You have to simplify that. You will get the answer 3 is to 2. Now, we will talk about this cube having a side 4 micrometer. Okay, so you will be given a diagram and they will ask you to determine the ratio of surface area to volume ratio. Now the answer is 3 is to 2. Fine for this. But what if the answer is given 1.5 is to 1? Even this is correct. Okay, so student may find that 1.5 is to 1 is wrong answer and they may ask that the correct answer is not mentioned in the options. If we simplify 3 is to 2 further, we get 1.5 is to 1. 1.5 times 2 and 1 times 2 is this 3 is to 2. Okay. So 3 is to 2 is also correct. 1.5 is to 1 is also correct. This is same as this ratio. Okay, we have been given a cube and side is 2. So that is ratio is 3 is to 1. What is true from the option? The cell surface area to volume ratio decreases when the cell size highly increases. When the cell size increase, surface area to volume ratio decrease. Okay, they are inversely proportional. So say for example, it's the largest cell. Okay, so when the cell size increases, surface area to volume ratio decreases. Khalid studying human skin cell under a microscope during biology class. He asked his teacher why cells are small. It's a very good question. You all also keep on asking why all the cells are small, why they are not large. So, which response does his teacher give him? So, what would happen if the cells are large? Remember, we know that this is a large cell and surface area to volume ratio is very low. If the surface area to volume ratio is low, the cell could not efficiently transport the nutrient. Larger cell could not efficiently transport the nutrient because the surface area to volume ratio is very low. Learning objective number 17, illustrate the main events of the cell cycle. Page number 226, figure number 4. So, to answer the questions based on this learning objective, you should know the diagram of the cell cycle. You should be able to label the structures or the parts of the cell cycle. And then you should know the events happening during each phase of cell cycle. So, broadly, cell cycle is divided into 3 phases. First one is interphase, in which the cell grows, DNA duplicates, and prepares for the nucleus division. This is the overall event happening during the interphase, which is the longest phase in cell cycle, interphase, where the cell remains for the longer time. Interphase is further divided into 3 sub-stages. Okay, the first one is G1, that is also called as gap 1. S is the synthesis phase, and G2 is gap 2 phase. In G1, cell is growing. and preparing to replicate DNA. Okay. So in the S, the DNA replicates. So what happens in the G1? G1 prepares for the DNA replication. In the S, the cell copies its DNA. Okay, various paraphrases are there. In S, what happens? The cell copies the DNA. DNA gets replicated, DNA gets duplicated. These are all paraphrases you should be knowing. Synthesis phase, cell copies its DNA, then G2. What happens in the G2? In the G2, the cell prepares for the division of the nucleus, because we know that in mitosis, nucleus divides, right? So G2 is a phase prior to mitosis. Yani, in G2, the cell will prepare to divide the nucleus. Okay. And finally, in mitosis, the nucleus or nuclear material divides. And the last phase, that is cytokinesis. Look at the word cytoplasm. The cell cytoplasm divide and finally two cells are formed after cytokinesis. All right. Let's see the example questions. In which phase of cell cycle cell prepares to replicate DNA, that is G1. Okay. In which phase of the cell cycle cell prepares to replicate DNA. Now, the common mistakes student can do over here is by not reading the question well. So what can happen? Student can simply read replicate DNA and they will choose the answer as S and lose the 4 marks. Okay, read the question well. In which phase of cell cycle cell prepare to replicate the DNA? We know that replication of the DNA takes place in the S and preparation starts even before S, that is G1. Right? Now, let's see the question. The stage where the cell prepare for mitosis. So this is mitosis. So which phase prepare for mitosis? The phase before mitosis, that is G2. What happens in cytokinesis? The cell's cytoplasm divides. Cytokinesis, cytoplasm, remember. So which of the following event occur during interphase? Interphase is a growth phase. Cell grows in size, DNA replicate, it prepare for the nuclear division. Okay, the same question repeated. Which phase prepares for the DNA replication? That is G1. Longer period of time, the cell remains in which phase? In interphase. It's the longest phase in cell cycle. Cell divides into two equal daughter cell in which stage? In cytokinesis. So that is denoted by C. C here. Yani complete division. S, here they haven't mentioned, so we need to identify where does S phase comes. That is in the place of B. Learning objective number 18. And infer the difference in the rates of cell division between cancerous and non-cancerous cells. So you can see these are the normal cells growing and these are the cancer cells. So we should know what is cancer. Cancer is the uncontrolled growth and division of cell. Okay. Moment you read cancer, the next thing what you should say is uncontrolled growth. Okay. Cancer results when cells stop responding to the controls of the cell cycle. Cell cycle have controls. If you remember the cyclin and the cyclin dependent kinase. Okay. So if they fail these controls, then what can happen? The cell cycle goes wrong and it continues to form a cancer cell. Some cells go through apoptosis. another important word. Apoptosis is a process of programmed cell death. Remember apoptosis with programmed cell death. And cancer with uncontrolled growth. Cancer is an uncontrolled growth of cell. Apoptosis is a death of cell. Okay. Apoptosis also occurs in cells that are damaged beyond repair. And the cells which cannot be repaired, including the cells with DNA damage that could lead to cancer. So in a way, apoptosis is good for us. Apoptosis can help to protect the organisms from cancer by killing them. Cancer is caused by mutation, change in the DNA. Mutation yani change in the DNA. The substances which are causing the cancer are called as carcinogen. Carcino means cancer. Gen means generating. Cancer generating or cancer causing agents or substances are called as carcinogen. Example questions. Focus on the keyword. Programmed cell death is apoptosis. Now, which is a part of the teacher's explanation? A biology teacher explains to students what cancer is. I told you, when you read the word cancer, it is uncontrolled growth and division of the cell. When the body cells divide out of control, look at this, the word is been paraphrased. Instead of writing uncontrolled growth, they wrote out of control. Both the meanings are same. Uncontrolled, out of control, both are same. Substances which cause the cancer are carcinogen. Not correct for cancer. Cancer cells have control over the cell cycle. No, we said uncontrolled cell growth. Learning objective number 19 from page number 227, figure number 5, expecting the figure to come in exam. Now here we can see these are the homologous chromosome. Chromosome at prophase stage on page number 227. The chromosome in prophase stage is sister chromatids that are attached at the point which is called as centromere, not centriole. This is centromere. Remember, the chromatids are attached to each other by centromere. Chromosomes are shaped like X. So this is X. Chromosome consists of sister chromatids that contain identical or similar copies of DNA. Sister chromatids are attached at the centromere of chromosome. So by now you must be very much sure about the structure which joins the sister chromatid is called as centromere. What is correct about the prophase? It consists of sister chromatids. Why are chromosomes visible as X-shaped structure? Because they are duplicated sister chromatids and which are joined at centromere. Learning objective 20 from page number 239, figure number 18. Now looking at this picture, you must be knowing that it is for meiosis. Meiosis 1 and meiosis 2. Now, meiosis is a type of a cell division that reduces the number of chromosomes to half. So it is called as a reduction division. It helps in the sexual reproduction for producing the gametes. What are the gametes? Male gametes, sperm, female gamete, ovum. Crossing over, or we also call this as synapses. It's a process that occurs at the prophase 1 stage. Remember crossing over or synapses and prophase 1. In meiosis 1, during which chromosomal segments are exchanged between the pair of homologous chromosomes that increase the genetic variation. Okay. What stage of meiosis genetic variation occur? That is anaphase 1, where the chromatids are being pulled away to different poles. Okay, which of the following is correct regarding mitosis? Only once the division take place. Mitosis, not meiosis. So read the question well, guys. Each question carries 4 marks. I'll just make this question bigger. Okay, this says that what is prophase 2 in the picture? Prophase 2. So prophase always comes before metaphase. This is prophase 2. Figure number 6. Prophase 2. All right. So next question. Now looking at the figure, you need to identify what is this. This is prophase 2. You can see there are 2 cells. Okay, and these chromosomes, they are already crossed over in prophase 1. And in prophase 2, you can literally, in the exam, you will see the color of this chromosome would be mixed, red or blue. In the same chromosome, the part of the chromosome would be colored in different colors like blue or red together. So this is prophase 2. Next. Now, look at the diagram. In the question itself mentioned as equator. So no doubt you can choose metaphase, but which metaphase? Is it metaphase 1 or is it metaphase 2? I am confused. Now it's very easy. Your confusion will be gone away if you understand metaphase 1 is when the homologous chromosomes are together. If you will go back, you can see this is metaphase 1 where the homologous chromosomes are paired together. Whereas in metaphase 2, you can see the single chromosomes are aligned at equator. Single chromosomes. Okay, they are already crossed over. Okay, they are denoted, they are represented by 2 colors. Okay, they are at the center, equator in the middle. Single chromosomes. Here you can see homologous chromosomes aligned at equator. This is metaphase 1. So this is metaphase 1. Now here, which stage of meiosis is, this is also metaphase. Now here you might not have given as equator, that's okay. You can see they are aligned in central line. Here they have directly mentioned equator. Here they mention in the question. Here in the diagram, this is all for metaphase. Why metaphase 1? Why not metaphase 2? Because they are in pair together, homologous chromosomes, so metaphase 1. This is anaphase 1, and why not anaphase 2? Again, in anaphase 1, you can see the chromosomes are moving away from each other. Again, going back to your diagram. This is your anaphase 1. Where the homologous chromosomes separate, they say chromosomes are separating here, anaphase 1. Whereas in anaphase 2, you can see chromatids are separating. Okay, so sister chromatids are separated in anaphase 2. So again here, the homologous chromosomes are separated from each other. Metaphase of meiosis 1 and meiosis 2. What's the difference between them? Again, homologous line up in the meiosis 1 and duplicated chromosomes line up in meiosis 2. Learning objective number 21, compare and contrast cytokinesis in plant and animal cell. Page number 230, figure number 10. So there are high chances of having the figure or a diagram in the question. Looking at the shapes of the cells, you can make out which one is the animal cell. Look at these round cells are animal cells. The geometrical figures like square or rectangle, these are plant cells. Now, in animal cell, cytokinesis begins with a furrow. Now, furrow is the connection between these two cells. These are animal cells, okay? Furrow is actually a pinch over here, a pinch formation over here. This is called as furrow. And eventually it splits the two cells apart. Here, plant cell build a cell plate that divides the cell into two daughter cells. So remember, for the animal cell, it is furrow formation, and for the plant cell, it is cell plate formation. Very easy. For this learning objective, you just have two diagrams. You have to remember animal cell with the furrow formation. You have to remember cell plate for the plant cell. Now, look at this. Cell plate is for the plant cell. So what do you describe this cytokinesis in this diagram as? Now look at the trick over here. Students can make a mistake if they won't read the options properly now. A cell plate forms in the plant cell. All right. There is an option which starts with the cell plate, but it says animal cell. That is why I'm telling you, read the question and option both well. Imagine if this cell plate form animal cell is in the first or the second option, and the student ignores reading third and the fourth option. Because moment he sees the cell plate, he chooses the first option with the cell plate. Don't do this. Read it well. Now this is animal cell and the furrow is here. Furrow forms in animal cell. Again, the similar mistake. Here the furrow forms is there. So the student can pick up this quickly without reading the all options, thinking that, okay, I know this. I read it well. I read it many times. I am very confident. This is furrow. I will choose it. But a student hasn't read it completely. He ignored this one, plant cell. Furrow is always in the animal cell. Why do plants cells form a cell plate instead of furrow? Because plant cells have got a rigid cell wall, okay, very hard cell wall. It doesn't form that pinch same like the animal cell. That is why it forms a cell plate instead of furrow. Which sentence distinguishes the process of cytokinesis in plant and animal? Cell plate in the plant and furrow in the animal. Learning objective 22, page number 225, figure number 3. Figure number 3, definitely this figure is going to come in exam because they have mentioned about the figure. What you should know from this, you should be able to label these all things. Parts of the structure. So this is the DNA ladder form structure. The round ball shaped structure, these are histone proteins. Histone proteins are wrapped by DNA. This is called as a nucleosome. Nucleosome is made up of two things, histone as well as wrapped with DNA. Then the loose form further is of the DNA is called as chromatin fiber. All right. Oh, sorry, the coiled form of the DNA is called as a chromatin fiber. It goes on compiling or coiling, and finally it forms the chromosome. So this is the relaxed form or a loose form. These are histone and a DNA, which is called as a nucleosome. Now this again coils tightly to form a chromatin fiber. This chromatin fiber further supercoils, tightly coils to make a condensed, super condensed structure called as chromosome. These are sister chromatids which are attached to each other by a structure called as centromere. Okay. The formation of chromosome. The negative charge DNA coil around the positively charged histone protein. So DNA and histone together to make the nucleosome. The nucleosome group together into chromatin fiber, which finally supercoil to make chromosome. Chromosomes consist of sister chromatids. They are identical or similar copies of DNA. Sister chromatids are attached in the center by the structure centromere. Correct about chromosome. It's a condensed form or a structure visible during mitosis. Which phase do you remember? After interphase, prophase. So in prophase, we can clearly see the chromosome. Now, relaxed form of DNA. Which is a relaxed form of DNA over here? That is chromatid. Chromosome is a supercoiled structure. Chromatid is a coiled structure. Chromosome again is a coiled structure. So loose form, relaxed form is chromatid, which is not the DNA. Interphase is not a form of DNA. Rest all these are the part of DNA or not. Which of the following is not a form of DNA in the nucleus of a cell? These all three are related to DNA. Interphase is a part of a cell cycle. Not correct about the chromosomes. The chromosomes are relaxed form of DNA. This is not correct. Chromosomes are the supercoiled form or the condensed form of DNA. A few more questions about labeling. They are asking you to identify the letter which represent nucleosome. So you should know thoroughly about this diagram, starting from DNA, histone, nucleosome. So C. Histone is F, sister chromatids E, and chromatin is D. This one. So once again, revising the structure: DNA, histone, nucleosome, chromatin, sister chromatids, and this is centromere. Question number 23, page number 228, figure number 6. This is about mitosis and cytokinesis. Now, because figure is mentioned, figure can come in exam or individual phase can come in exam. So the question says, when does the chromatid separate away? Ana, that is anaphase, so B. Equator, middle, meta, so it is E. So a phase in which you can see the chromosomes. Chromosomes are first seen in prophase. Okay, these are all indirect questions. Letters are mentioned and what events are happening, those are described, and you need to identify the phase where this happens. Right? Now there is a question. You need to identify this phase. This is a telophase, which is the last phase in mitosis. Again, the similar question number 24, page number 240, differentiate between mitosis and meiosis. The main function of the meiosis is to produce the gametes, that is sperm and egg, and ensure the genetic variation. What type of cells are involved in mitosis? Those are all somatic cells or body cells. For example, skin cell. So instead of writing somatic cell, they may write sperm, egg, or skin cells, or liver cells, or body cells. These are cells in which the mitosis happens. What is the key difference between mitosis and meiosis? Mitosis produces similar cells and meiosis produces genetically different cells. Learning objective number 25, page number 244, table number 2. Definitely we are expecting this table to come in exam. Either the full table or individual structure to identify the phenotype. Okay. To understand this and to answer this question correctly, to get full 4 marks, we must know the concept. Now, cell division during which the sister chromatids fail to separate. What does fail to separate? Sister chromatids. Okay. This is called as non-disjunction. So here in the diagram, you can see. So here, instead of the chromatids like this, they are separating, chromatids are separating. Here, the chromatid has not separated properly. So the whole chromosome has gone at one nucleus. Okay, this is called as a non-disjunction. So what can happen because of the non-disjunction? Non-disjunction can result in extra copies of certain chromosomes or only one copy of particular chromosome in the offspring. So here you can see this is a normal one diploid. But if there is an extra copy, so it can be a trisomy. Or if there is one chromatid missing, it would be 45 chromosome, that is monosomy, right? If the offspring have 3 set of a same chromosome, called as a trisomy. If the offspring have only one of the particular chromosome, it is called as monosomy. Down syndrome is an example of trisomy 21, where there is an extra chromosome found on the 21st pair of chromosome. An individual with the Turner syndrome, there is a Turner syndrome here, this one. Only one X chromosome is there. Sex chromosome, only one is there. X0. Only one is there. This results into Turner syndrome. And individual with one extra sex chromosome will have a syndrome called as Klinefelter's, which is seen in the male. So you can see there is one extra X chromosome. This is a trisomy, which is called as a Klinefelter syndrome. So if you will observe this very clearly and carefully, it will be very easy for you to identify. Look at this, the first genotype XX is a normal female, genetically classified as female, whereas XY is genetically classified as male. So these two are very easy to identify, XX and XY. Okay. For the females, there is only one syndrome mentioned. Except from the normal XX genotype, there is only one phenotype which is mentioned here, that is having only one X chromosome, that is Turner syndrome. Okay. And if there is one X chromosome extra for the female, there is no phenotype effect. It is a normal female, lives a normal life, no phenotype effect. Similarly, in the male, if there is one extra Y chromosome, male chromosomes are XY. Genotype is XY. If there is extra Y, no phenotypic effect. Similarly, this is a female XX. If there is one extra X, there is no phenotypic change. Then, coming to this trisomy, now XY is a male. If there is one extra X, Klinefelter's syndrome. And the last one is 0Y, or only one sex chromosome, that is Y chromosome, results in death. Very easy. Y chromosome results in death. So what kind of questions we expect? So you will be given a diagram. Now how to identify this? This is XXY. This is a male. XY with one extra X chromosome. This is a male with Klinefelter syndrome. Okay, very easy. Male with Klinefelter syndrome. Similarly, this one is a female, XX, with one extra chromosome, so no phenotypic change. Only one X chromosome, this is Turner syndrome. Now, again, this is a male XY with one extra Y chromosome. Similarly, like this, females have no phenotypic exchange if one extra chromosome. Males have one extra Y chromosome, no phenotypic change. Which of the following phenotypes can be identified? You can see it's just one single Y results in death. This is normal male genotype XY. Why does non-disjunction occur? So the sister chromatid, if they do not separate, what happens? The non-disjunction happen. What could explain a human karyotype showing 47 chromosome? 47 chromosome is one extra chromosome, that is trisomy. If they ask you a question for 45 chromosome, any one chromosome less. So we should normally have 46 chromosome, right? If one extra, it will become 47. This is called as trisomy. If one chromosome is missing or less, for example, 45 chromosomes are there, this is called as monosomy. Mono yani one, tri yani three. So finally, we have come to end of this revision. So how to write the exam? You need to read the question well and highlight the keywords. Read all the options and choose one suitable answer. Use only blue pen for your exam to circle the option. This is very important. Please do not do a lot of correction. Yani, for example, some students are choosing two options or maybe three options and then cutting it and then finally writing it there. This is my correct answer. Please don't do this. Select one clear-cut answer. Do not use the whitener. Do not be in a hurry to submit the exam. Please review all questions before submitting. Here I would like to give you advice. Some students change their answers at the end. You should have enough confidence why you have chosen that answer. Otherwise, we find that there are some students, their answers were correct. At the end, they have changed it. Each question will have minimum 4.30, yani 4.5 minutes. Manage your time accordingly. All the best. Thank you. And great job, everyone. That brings us to the end of our term 3 revision session. Remember, understanding the concept is more important than memorizing facts. Go over your notes, practice with past papers, and don't hesitate to ask questions if you need help. You have worked hard all term. Believe in yourself and give your best in the exam. Wishing you all the very best. Stay focused, stay positive, and good luck. This is Miss Abeda Peerzade from Mother Girls School, Fujairah. Wishing you all the best for your term 3 biology exam. I wish you all get 100 out of 100 marks.